I'm Siti Othman from Laboratory of Animal Behavior and Conservation, Nanjing Forestry University. Today I would like to share a research entitled In Absence of Sexual Selection, Toads Release Calls Can Signal Evolutionary History. And to begin with, I would like to introduce the model system that we have used for this landscape level of phylogeography, and we have selected the Mongolian Toad, Strauchbufo raddei, as our model system, as this species is one of the most widespread species in Northeast Asia. And because of that, this species also inhabiting diverse biotopes or biomes in East Asia, which is ranging from the grassland of the Gobi Desert, as well as the joint mountain of the Qinghai Tibetan Plateau. And this species also inhabiting the river basins in the Northeast Asia. And besides, this species also inhabiting several elevations. For instance, in Western Chinese mountain, this species can reach as high as 3,000 meters. And first, we have to know what is the calling in anurans and its relation with sexual selections. And for the social context of the emission, the anuran vocalization can be grouped into three major categories, which are the reproductive call, aggressive call, as well as the defensive calls. And vocalization that mediated these two components is mostly linked to the sexual selection in frogs and toad. And what are those two components? They are vocalization that mediates the male-male competitions, as well as the vocalization that mediates the female choice for her mate. And what is release call in toads? So release call it's grouped into the reproductive type of call, but release call can be emitted by male and female when they are tentatively impacted. Here is the by a male individual, and here is the example of the position and plexus position during the mating between male and female toad. And male will use the release call as a diagnostic signal to differentiate the individuals, whether the individual that they want to mate, whether it's a female or it's unreceptive males. So hypothetically, release call are not evolutionary diverging due to its function as diagnostic signal, and it has to be conserved among conspecific species, as well as sometimes it can be conserved among congeneric toads. And to address the research question number one, it's about the how diversity of landscape can impact the genetic diversification and phenotypic traits of Strauchbufo raddei. And to address that, we did a sampling, genetic and acoustic sampling of Strauchbufo raddei. So we sampled 147 buccal swabs of Strauchbufo raddei along the Mongolia to Russia. And in the Mongolia, the westernmost range of distribution that we did our sampling are located in the Selenga rivers, as well as the western margin of the Hentian Mountains and close to the Trans-Baikal, while the easternmost part consists of the Strauchbufo raddei that is distributed at the Amur River basin, close to Lake Kankal. And for the acoustic sampling, the release call, we have sampled the release call distributed between two margins of Hentian Mountain, the western margin as well as the eastern margin. So overall, we have like 29 release calls from males. And we proceed for the molecular works, where we collect the DNA as well as we amplify the sequence, which is the empty DNA sequence. And we did the data analysis using the bioinformatic framework that consists of phylogenetic, phylogeography, and population genetics. And to answer the first question, which is the impact of landscape's variability to the phylogeography, so we analyze the phylogeny of Strauchbufo across its entire distribution, and we recovered 7 clades overall for the species across its entire distribution in East Asia, where the 7 clades clearly can be separated into 2 major groups, which we call it as Northeastern originated group, or group 1, represented by clade 1 until 5, and we can see the distribution in the map here, 1 until 5. And the second group is represented by the Southern originated group that restrict to the clade number 7 and 6. And we want to highlight the Southern originated group consists of the Strauchbufo that is distributed along the Sichuan basin to the Gobi Desert, as well as the western margin of the Hentian Mountain, close to the Trans-Baikal. And interestingly, we did the molecular dating for the same CR fragments of S. raddei across its entire distribution. And what we found that the basal lineage for the 2 originated clades, the 2 originated groups, indicate that the Strauchbufo emerged in East Asia at least since 10 million years ago. And after that, subsequently, the 2 groups separated, the Northeastern originated clade as well as the Southern originated clade. They have been separated ever since 8 to 6 million years ago. And to highlight the impact of landscape variability, where the Strauchbufo that also distributed across the East Asia were at least distributed in 5 diverse biomes. So we assign the Strauchbufo towards to their specific landscape across the species range, and we infer the gene flow rates for each clade of raddei that have been assigned to their landscape. And we could see that there are symmetrical gene flow in 3 types of landscape, where the Strauchbufo that distributed in the steppes or grassland, symmetrical gene flow to the river basins, as well as from the Strauchbufo that distributed in the river basin to the canyon, as well as canyon to the steppes. And we could see that there are restricted gene flow in the Mongolian Toad that distributed in the plateau landscape, as well as the lowland forests of the northern Yangtze. And clearly the differences in the flow, in the gene flow, indicate that the landscape did impact the migration pattern of the Mongolian toads. And to answer the second research question is, can phylogeography influence the release calls of anurans? So here is to answer the previous hypothesis that release calls are not evolutionary diverging and they have to be conserved among conspecific species. And to address the second question, we focus on the northern limit range of some Strauchbufo, means that indicate by the Mongolian and the Russian clades. And here we also provide evidence that the northernmost range of S. raddei can be distinctively grouped into 2 clades, which are clade A, that distributed at the eastern margin of Hentian Mountain, as well as clade B, that distributed at the western margin of Hentian Mountain. And these 2 distinctive clades also having a similar pattern with the 2 distinctive clades that we recovered for Strauchbufo across its entire distribution. It's mean that one of the both clades were having different origin. We say clade A originated from the northeastern origin, and clade B originated from the southern originated clade. And And we also look at the past population dynamic to infer whether there is a pattern of glacier refugia in the clade distributed at the Western Hentian Mountain. And what we found is a significant differences between the past population dynamic of clade A and clade B. For instance, for the clade A here, you can see that the past population dynamic since 250,000 years ago, clade A is having a very stable population growth, and what's the present time, it's inclined and the population get reduced a little bit. Meanwhile, for the clade B that located at the western side of the Hentian Mountain, we could see the past population dynamic since 40 years ago, showing a gradual increase up until the present time. So it's indicate that the pattern of glacier refugia and the range expansion that occurred from the southern originated clade to the western Hentian Mountains, it's a positive pattern. And for that, we also proven that there is a phylogeography that influenced the structure of northern limit range of the raddei between the clade A and clade B. We also want to look whether the release call reflect the same patterns. And here what we found when we analyzed the acoustic data for the clade A and clade B Strauchbufo raddei, we found a significant difference between the three variables of the release call, which is call durations, rise time, and fall time. They are showing that clade A is having longer time compared to the clade B. And this pattern of call, that's the patterns of release call that are significantly different between Strauchbufo raddei belong to the clade A and clade B, indicate that the release call is following the genetic structure pattern as well. And it's also maybe influenced by the Hentian Mountain there, that I guess it's a genetic barrier, as well as the phenotypic trait have been influenced by the natural barriers. So as the take-home message, what we want to highlight is that landscape variability did affect the structure of the phylogeography of Strauchbufo raddei, and phylogeography or evolutionary diversification did impact the structure of release call, which is contrary to the past hypothesis. And I would like to thank my collaborators and funder for this project, as well as thank you. And yeah.